Hello and welcome back to Something About Science. I'm your host, Danielle Ellis, and today we're talking all things SLAS 2025. This year's conference brings together the brightest minds in science and technology to explore groundbreaking advancements in laboratory automation, innovation, and analytics. Joining me are the educational program chairs, Dr. Herve Tiriac from the University of California, San Diego, and Dr. Meiye Wu from 10x Genomics. They've been instrumental in curating this year's educational program, so it's an absolute pleasure to have them with me so we can talk about the educational program in more detail. Without further ado, let's hear from them. Thank you, Herve and Meiye, for joining me today. It's an absolute pleasure. We just want to get started by talking through, how you got involved with SLAS and how you became educational program chairs. So, Herve, if we can start with you.
Sure, I'm happy to. So I'm Dr. Herve Tiriac. I work at the University of California, San Diego. I'm in the Department of Surgery at the Morse Cancer Center, but I work as a scientist in the laboratory and I study pancreas cancer with a special focus on 3D biology and organoids, patient-derived models per se. And this is really what brought me to SLAS. So in 2019, I was invited as a speaker to speak at a symposia that was focused on 3D biology. And that went really well. And that kind of translated into me helping organize the same meeting over two other sessions, two other years, once as a session chair and another time as a program committee member. And then I continued my journey with SLAS because I really enjoyed it. I helped co-chair a track focused on precision medicine for the main meeting. And then the following year, I tracked the chair. And now I'm really thrilled to be one of the co-chair of the program for SLAS 2025 with Dr. Meiye Wu.
Perfect. And that's the perfect transition for Meiye.
Hi. So my name is Meiye. I am currently working as a director of manufacturing at 10x Genomics. It's a cutting edge single cell analysis multi-omics company. And my involvement with SLAS started back in 2013 when I was a finalist for the Innovation Award. And I was working on single cell analysis at the time for my Ph.D. So since that was my intro to SLAS, and then from there I had the opportunity to be a student poster judge. And after that, I was the judge for one of the panel judges for the Ignite Startup Award. And last year, I was they invited me to be the track chair for the omics track, and that went really well. So this year, I'm honored to serve with Herve as a co-chair for SLAS 2025.
Perfect. Thank you both. And for those who aren't quite sure, what is the role of the educational program at a conference like SLAS? What does it aim to do for attendees?
So, the goal is to have a breadth of topics to be covered, both things that have been covered in previous years and are being updated, and those are core parts of the SLAS mission. But it's also to cover new topics, emerging topics, which we'll discuss later, I'm sure, and to give everybody a breadth of knowledge that helps them sort of move forward with their career.
Yeah, and on that breadth of knowledge, what are, SLAS has 8 educational tracks for 2025. What are those tracks? Perhaps if we start with you, Meiye, we could take the first 4.
Okay, yeah. So very exciting. So we have our new modalities track, which is a double track this year. And that's kind of what's in the forefront, what's innovative. And then this year we're going to have emerging technologies in the drug discovery space, including RNA targeted therapies, as well as strategies to go after undruggable targets. And we'll also discuss the future of protein therapeutics. So that's really exciting. And then we also have a track called micro nano technologies, and that's just what it sounds like. So these are tiny micro nanoscale, devices that scientists are using to study single cell proteomics. We're also going to cover topics in the latest 3D printing technology for bio applications. and just to see, what the big impact they can have using these tiny technologies. Our really recent huge track is in AI and data science. And that has gotten a lot of interest for SLAS in the last, couple of years. And it's, as we all know, the AI has just been a revolution. And so we, really apply that in the context of drug screening. So we use it beyond just looking at, what they can do for, in the context of high content screening, and also how we use it to analyze data in the omics landscape. And along with that, there are a lot of infrastructure improvements that needs to be made to accommodate all this computing power from AI. So we will cover that as well, just the challenges in data integration and engineering, and how we can create real value. And finally, the omics track. So because omics did, really well last year, so we decided, based on attendee feedback as well as the programming committee, that it's a double track this year. So really what got spun out was the spatial omics track. And so there, I think that we get to see the latest and greatest in spatial omics, which is a combination of looking at multiple analytes, multiple types of biomarkers from the same cell. And add on top of that, you are looking at a spatial context where the cell is still residing in the tissue. So that preserves that 3D, microenvironment, and that gives you a lot more information to, assess the biology, either in a healthy tissue or in, aberrant, like disease tissues. So that's actually really exciting for 2025.
That reminds me of the recent edition of Nature that I think it went live on Halloween, so the 31st of October. It was about the tumor cell atlas, and I think the phrase they used was spatial awareness.
Yeah.
I think was it spatial transcriptomics, or it was some sort of spatial omics. So it's very much something that is at the forefront of the scientific community at the moment. I know you said that this decision was based on attendee feedback. Are you aware of the wider scientific community as well, and sort of attracting people that maybe haven't been to SLAS before with this track?
Yes. Yeah, I'm glad you brought up the atlasing, because that is a huge, I guess, endeavor. There are several global efforts to one of them is the Human Atlas Project. So we're trying to, put together atlas basically cell by cell for the entire human organism. And that's super exciting, because that's going to lay the foundations for so much to come. And SLS is just really getting in the front of that. I think it's great.
Yeah, and that's where these, important conversations between decision makers can happen is, around this track at SLAS. Herve, if we move on to the other 4 tracks, do you want to talk us through those?
Yeah, absolutely. So the first track I'll discuss is the asset development and screening track. So this is a track that's coming back from previous year. It's been quite popular. And it really delves into the breakthroughs of how we develop and screen assays. This track is all about the innovative ways in which scientists are finding new biological and active compounds in screens. And we're going to really discuss real-world success stories where novel technologies have made screening campaigns more effective and efficient, which is really good for the audience to kind of see how all of this is applied in different industries and academic centers. And if you're curious about how cutting-edge assays are driving drug discovery forward, this is a good track to attend. Another track that's coming back that has 6 sessions, I believe, is the automation technologies track. And this is, again, about how automation is continuously improving the laboratory. In this track, we're really focusing on new tools and technologies that are making high-throughput screening and lab processes faster, better, and often easier for people. There'll be a lot of discussion around how advanced robotics and smart software with AI, for example, are improving how we collect and analyze data. And this is really how technology is leveraged for the betterment of science. Then 2 shorter tracks close to my heart. First, the cellular technologies track. This is a track that really looks at how we make models and how we apply these models to screens. We're going to be talking about organoids, organ on a chip, and many other models that are changing how we're studying disease and develop treatments. We're going to hear from experts who are using these technologies to make drug discovery and preclinical development possible. And it's a really good look at how we're trying to advance more physiologically relevant models to the lab, both in academia and again in industry. And then a new track for this year, screening applications in biomarker diagnostics. This track will explore how emerging screening technologies and biomarker diagnostics are transforming healthcare. There's going to be discussion about how we need robust assays to improve diagnostic screens. It's a big area of need. And how all of this impacts patient care. There will be also discussion about how algorithms that are looking at these biomarkers are really critical to be evaluated because they're enhancing disease diagnosis, but they also, streamline patient outcomes and really affect the way we treat the patients. So it's really important for those algorithms to be well considered. And it's really an exciting glimpse into the future of medicine. I really like this general track. This is the evolution of the precision medicine track, and I hope it's well received this year.
Yeah, on that very last point of it being a continuation of the precision medicine track, am I right in saying that there was sort of two tracks combined?
Yes.
I was just going to say, how did you come to that decision to combine the two? Was it like me was saying before, was it attendee feedback?
Yeah, so this is the combination of both precision medicine and diagnostics, which was one track, and advanced in bioanalytics and biomarkers, which was another track. And you can see just from the title of those two tracks that there's a lot of interdependence, biomarkers and diagnostics are pretty much the same thing. So we really need to acknowledge at SLAS the convergence of all these fields and really provide just one track for people to attend so they don't have to split their attention. Or maybe miss a really interesting talk that they would be interested in. So I think it made total sense to kind of combine these two into a track and really provide the SLS attendees, one unified vision for precision medicine, diagnostics, and biomarkers.
Thank you. Moving on to how you sort of curate an educational program now. I'm sure from people on the outside who have no experience of this, they just think, how can you take this wide variety of possible topics and curate it into, an impactful educational program? On that, how do you prioritize these topics? Because I imagine it's like picking your favorite children. Like it's really, really hard because there's probably so much interesting stuff that you could include, but you can't include everything.
Yeah, that's right. So we have a carefully curated educational program that's around the main track, scientific tracks that we just discussed. And we build it up by really closely monitoring the emerging trends, like we discussed earlier, and listening to the scientific community's feedback and the previous attendees' feedback. And so, for example, we were just talking about AI. So we have a whole course about practical introduction to data science models using AI and generative AI. And this is new for this year. And there's another course that's very similar that's called Nexus XP, which is planning your future lab, and also talks about sort of how to integrate, new technologies in the laboratory of tomorrow. But we also don't want to, go too far away from what we've done in the past. So we still offer the fundamental courses that we've offered in the past, such as an introduction to mass spectrometry and its application within drug discovery. A lot of courses on automation and things like a little bit newer things that are really interesting, but I think should have been mainstays in previous years, like exploring targeted protein degradation concepts and application. And all of these kind of make sure that we cater to both returning audiences that want to see new things, but also train the new generation with the same educational program that has helped previous attendees sort of forward their careers.
Yeah. And am I right in saying that Nexus IP is new for 2025?
Yeah. So Nexus XP, I believe, is new for XP, 2025. Sorry. And the AI course that I mentioned, the Practical Introduction to Data Science Methods, is also new. And I believe the protein degradation course is also new for this year.
Okay, so lots of new stuff, but also, returning attendees will be familiar with certain aspects of the program as well.
Absolutely.
Yeah, brilliant. And then talking about the trends you just mentioned, What are some of the trends that you are both most excited about for this year that you've seen and that have been implemented into the program?
So the Nexus XP is actually super exciting. I mean, this concept is very cutting edge. So it's the design, build, test, iterative loop that everyone's talking about. You know, you design something using AI, design a set of experiments, and you have automated testing, and then that cycle just repeats itself, iteratively. And so I think, Leslie, our scientific director, really did a great job of pulling that together, because just that concept itself of how do you even do that at a conference? And this is going to be I think, people's choice. It's going to be a competition. I think there are companies that signed up to do this demo, and it'll be exciting. They have their own space, and attendees get to go in there and experience different ways of doing this kind of, design, test, iterative process, and see how we can take that concept, actually apply it in a screening application. So I'm really personally, that's where I'm going to go. I'm going to see like, hey, how is anyone implementing this? Yeah. So that's Herve. What about you?
Yeah, I'll do a shameless plug because there's my interest in 3D biology. There's actually quite a few courses that are focused on that. So we have one which is titled 3D Printing for Scientific Applications, which is a little bit outside my expertise, but really brings in the importance for 3D and in building assays now. And then we have two assays that are very similar to two courses that are very similar to the stuff that I do, which is one of them is Level Up Your 3D Cell Culture. And the other one, which I co-lead, is How-To Toolbox for the Development of Physiologically Relevant 3D Models. And obviously, we believe, and I strongly believe that those are really important for the betterment of assay development, as we move towards more physiologically relevant models.
Perfect. Thank you. SLAS is really passionate about both curiosity and innovative thinking. How does the program sort of, ensure that attendees have the opportunity to, be curious and think innovatively? Because, we want to educate people about the science we've done so far, but, these attendees are going to be the scientists of tomorrow making the new discoveries of tomorrow. How do we implement them with the skills to be able to do that?
Well, we have all those classes they can take.
Yeah.
There's really the broad range of classes, I think, is really impressive, and also that applied aspect of it. It's not just theory, but people actually get to touch, and they get to experience, and they get to learn from experts who are working in the field. I think that's a very important aspect of SLAS, because it's not just a scientific academic conference. It's also a trade show. And it's the premier trade show for automation, lab automation. So it's that intersection between the industry, the academia, and the in-between is really where I think really prepares some of these, junior scientists or aspiring engineers to think outside of just one, their own little, corner of academia, right? So it's really opening their minds to different people, different technologies, and what's coming out of academia as well as the startup scene.
Definitely. And I suppose it can't be underestimated as well, having, people talking about spatial awareness earlier, having everyone in the same place at the same time. We've come off the back of COVID-19, and obviously trade shows like SLAS had to be on pause, but having everyone back in the same place, having, conversations in person, it can't be underestimated.
If I can comment on that, one of the things that I love about SLAS is that even though it's a really big trade show and it really feels really big, you do have this feeling of a small meeting after sessions and during the courses where you really get a lot of personalized time with the speakers and the presenters and the lecturers. I often, when I present or when I teach, I often call on the audience to specifically ask questions or comment on things that we're discussing, and it really has this vibe of discussion as opposed to just being talked to, which I really appreciate.
Definitely, that sounds really, really valuable. to be in such a big place and a big, like you said, it's a trade show as well as a conference, to get that small meeting feeling is really important. Thank you. Looking, what are your hopes for SLAS 2025 in terms of, when the show's over, when everyone's gone home, what do you hope people have taken away from SLAS?
Well, I mean, speaking from my perspective, is that I hope everyone leaves SLS the same way that I do every year, is that we've had fun. We had fun meeting each other. We had fun looking at new technologies. We had fun listening to, great science. And also I hope they leave inspired with some, new ideas. And just having been under the same roof with, to such high caliber scientists and engineers, as well as the cutting edge vendors out there making, that can actually make ideas happen. And I hope they leave thinking, hey, you know what, maybe I do want to start a company with, the people I've met, or maybe, and also I think it's really important that the networking aspect, I hope they leave having met some, potential partners in the future, potential mentors, and set up, relationships that will last beyond the SLAS conference.
And Herve?
Well, 100% what Meiye just said. I think the networking is really important. For me, a successful meeting is one that I come back with being surprised by a couple of things that I didn't know that inspire me to do new things or start new collaborations. And this often happens at SLAS. So I think if that happens for most people, it'll be successful.
Brilliant, thank you. And we're just going to end on one little fun question. And the question focuses around SLAS's love of curiosity. So I was just wondering, when you were young scientists or maybe even young children, what were you curious about within science and what are you curious about today? For instance, I loved space and dinosaurs, but that's like the typical child response, isn't it?
Oh my gosh. As a child, I wanted to be a fashion designer. Did not go. So, curious about
college student.
Yeah. Oh, I'm such a generalist. I was just curious about what can I make? Like, how do I take that great idea and how do I make it happen? Like, what's between idea and reality? Like that.
That's the fashion designer in you. What can I make?
and how do I make it look good, right? So I think, it's kind of hard because not everyone started out. You don't always end up where you think you want, right? So it's that journey that's in the way, that happens along the way. I think is super interesting. But what you do realize is that things are really interconnected. Like music, math is connected, art and science is connected. And that at the end of it, when you're really trying to make something and you realize you actually need all the aspects, not just the science, not just the math, but you also have that human aspect. Because everything you design, every piece of equipment, every instrument, it does have a human machine interface. And a lot of thought goes into just designing that and how would that piece of equipment, how would that technology work in a researcher's hands? Is it actually going to give you the kind of, usability and the impact that you're going to have? So I think it's a very hard question, right? That's assuming you wanted to be something when you were little and then, you repeat that. There's some people are lucky that they can do that. But I think for me, it was just meandering, career path.
But that's good that you were just curious about everything.
Yeah. How do I be smart and look good and, have fun?
That's all you need.
That's just a successful life.
Yeah. What about you?
I'm working on it.
I mean, a little bit corny, but my interest in biology really was initially genetics. And that's what I went to college for because I was so passionate about it.
Yeah, me too.
The more I learned, oh, awesome. The more I learned about it, the more I kind of discovered that it wasn't as interesting as I thought it was going to be. First of all, it's a lot more complicated than just dominant and recessive genes, which I thought was the whole game initially. So what I'm kind of evolved into it is sort of like what is beyond genetics. So not going to bore everybody with it, but like, epigenetics, transcription, whatever, protein. It makes everything so much more complicated and so much more plastic. There's just no fast rules in biology. Almost everything is flexible, and really everything that I know is only such a small portion of it that I'm just humbled every day by what we don't know and what we don't appreciate yet. So I'm just trying to stay abreast of all the literature, and it's a continuous struggle, So my initial aspirations to be a geneticist, I've kind of been thwarted by the fact that it's just not good enough. You need to be an expert in so many other things to be able to make progress now. So it's a continuous struggle, but I love it.
Herve, you have to struggle because you know what? Oops, because I have no time either. Is that ChatGPT is great at summarizing
Oh yeah,
papers.
it is.
That's
It
always
is.
a struggle, right? If you've ever been in academia, it's just keeping up to date with
Yeah,
publications, and it's only gotten worse because there's just more
absolutely.
publications now. Like there's a lot more, open source journals, and just
Yeah.
in a just tsunami of new things every day, right? So I mean,
Yeah.
I think using that in our daily lives, I think would really help just to stay on top of things.
Perfect. Well, we've reached the end of my talking points, and I think that was the perfect end to our conversation today. So I'll just say thank you, Herve and me, for joining me today and having this insightful conversation. If people want to know more about SLAS 2025, they can, of course, visit the SLAS website where they can register.